Good morning. Myself Dr. K. Swathi, working as Associate Professor in Sri Padmavati Mahila Vishwavidyalayam, Department of Institute of Pharmaceutical Technology. So today's topic is qualification of HPLC from Pharmaceutical Validation, Semester 1, Department of Pharmaceutical Analysis. Contents is introduction, qualification, applications and references. What is qualification? Action of providing and documenting that equipment or ancillary system are properly installed, work correctly and actually lead to the expected results. Design qualification, installation qualification, operational qualification, performance qualification. So coming to HPLC, HPLC is a technique for disjoining, determining and quantifying each component in a mixture. HPLC is a form of column chromatography that pumps at high pressure a sample that is unlight dissolved in a solvent that is in a mobile phase through a column with the immobilized chromatographic packing material that is in the stationary phase. As the sample passes through the column, analytes having the strongest interactions with the stationary phase exit the column the slowest, meaning they exhibit longest retention time. In contrast, samples demonstrating little interaction with the column material elute quickly and are thus characterized by short retention times. The properties of the sample and the solvent as well as the nature of the stationary phase determine the retention time of the components. Design qualification. It describes the user requirements and defines the functional and operational specifications of the instrument. DQ should ensure that the instrument to be purchased have the necessary functions and performance that will enable for suitable intended application. So in this design qualification, that means before purchasing a new instrument. Installation qualification, a documented installation of new or existing instruments. Operational qualification, after installation, after major changes like repair, updates at regular intervals like risk based. Performance qualification, whenever the instrument is used, that is daily. Coming to design element, intended use, the example is analysis of drug components and impurities. User requirement specification for the HPLC analysis is up to 100 samples per day. Automated overnight analysis, limit of quantification 0.1%. Automated confirmation of peak identity and purity with diode array detection. Automated compound quantification printing of the components. Functional specification, pump, detector, auto sample, column, compartment, all should be checked. In this example is binary or higher radiate, UV or visible diode array 190 to 900 nanometers, 100 samples, 0.5 microliter to 5 ml sample volume, 15 to 60 degrees is controlled. System control data acquisition for signals and spectra, peak integration and other components. Coming to design element, operational specification. In this example is detector baseline noise less than 5 into 10 and 5 angstrom units. Sample precision injection volume less than 5% RSD. Sample carryover less than 0.5% RSD. User instruction, operation manual on paper, computer based tutorial. Qualification is that the vendor must provide procedures and services for IQ and OQ. Design element, maintenance. The maintenance, like vendor must deliver maintenance procedure and recommended schedule. Instrument must include early maintenance feedback for a timely exchange of most important maintenance parts. Maintenance procedures must be supplied on multimedia CD EOM. Training, vendor must provide familiarization and training. Installation qualification. Installation qualification establishes that the instrument is received as designed and specified. It establishes that instrument is properly installed in the selected environment and that environment should be suitable for operation of this instrument. Run of test samples verifies correct installation of all modules, electrical and fluid connections. Equipment is compared as received with purchase order, includes software, accessories, spare parts and consumable. Equipment is checked for any damage. Hardware is installed following the manufacturer's recommended. Documentation checked for completeness of operating, maintains instructions of the standard operating procedure for testing, safety validation certificates and health safety instructions. The supplier instructions are read if there any The instruments are site switched on, ensured that all modules power up and electronic self-test performed. Before installation, obtain manufacturer's recommendations for installation site requirements. Check the site for the fulfillment of the manufacturer's recommendation. Allow sufficient shelf space for the equipment, SOPs, operating manual and software. During installation, compare equipment as received with purchase order. Check documentation for completeness. Check equipment for any damage. Install hardware. Switch on the instrument and ensure that all modules power up and perform an electronic self-test. Identify and make a list with a description of all hardware. Include drawings where necessary. Run test sample and compare chromatogram print out with reference chromatogram. Prepare an installation report. Coming to operational qualification, it is a process of demonstrating that an instrument will function according to its operational specification in the selected environment. It verifies that the HPLC system complies with key function and operational requirements as specified in the design qualification. In operational specification, the supplier must define exactly the conditions that must be observed with the verifying conditions. Before performing all other tests, first perform leak test. If it is failed, then most of the remaining tests will be failed. Test parameters and acceptance criteria. The first parameter is leak testing. Procedure: flow test by volume or weight per time. The limits must be plus or minus 5%. Baseline drift, that is ASTM, American Society for Testing Material method, E19 9.20 minutes should be followed. That the limits is less than 2 into 10 to 3 angstrom units. Coming to baseline noise, the procedure is ASTM method and less than 5 into 10 to 5 AU. And the same way, all the parameters like precision of injection volume, precision of flow rate, detector linearity, wavelength accuracy, temperature accuracy, they can be as per the procedures we have to test, and the limit should be maintained like this. The parameters and procedures and other limits is temperature precision, auto sampler carryover, mobile phase composition accuracy. They should be within the limits and it should be followed the standard procedures. Coming to performance qualification, it is the process of demonstrating that an instrument consistently performs according to a specification appropriate for its routine use. Important here is the word consistently. The test frequency is much higher than for operational qualification. Another difference is that performance qualification should always be performed under conditions that are similar to routine sample analysis. For a chromatogram, this means using the same column, the same analysis conditions, and the same or similar test compounds. Performance qualification should be performed on a daily basis or whenever the instrument is used. The test frequency of only depends on the stability of the equipment but on everything in a system that may contribute to analyze the result. For a liquid chromatography, this may be the chromatographic column or a detector's lamp. The test criteria and the frequency should be determined during the development and the validation of the analytical method. In practice, performance qualification means system stability testing, where critical key system performance characteristics are measured and compared with the documented present